Hello everyone. Today we will study about the back cross and test cross. I have already explained regarding the monohybrid and dihybrid cross. You have been also told about all the three laws of the Mendel's: law of dominance, law of segregation, and law of independent assortment. Now, as I have written here, back cross and test cross. What do you mean by back cross? The back cross is the cross of F1 hybrid plant to their parents, either dominant one or recessive one. You can see here with the example. Suppose the capital TT and this one is the small TT. This is for tall plant and this one is for dwarf. And what is the F1 hybrid? Capital T, small t. This is the hybrid plant and the generation is F1 generation. So back cross is the crossing of this hybrid plant to their parents, because the crossing is going to be made back to their parents, either the dominant one or the recessive one. So crossing of the F1 plant to their parents, either the dominant one or the recessive one, this is called back cross. And test cross is a type of back cross in which the F1 offspring, or you can say the F1 hybrid, is crossed to their recessive parent only. So back cross, you can write here back cross. Back cross is the cross of F1 hybrid plant to their parents, either dominant or recessive. And what is test cross? Test cross is the cross of F1 hybrid plant to their recessive parent. Have you got the differences between back cross and test cross? As I told you, back cross is the cross of F1 hybrid plant. This is the F1 hybrid plant to their parents, either the dominant one or recessive one, and especially the cross of F1 hybrid plant to their recessive parent. This is called test cross. Now there is a question that why the test cross is made? What is the importance of test cross? As I told you, test cross is a type of back cross. What is the significance? Overall, the importance of the back cross is to maintain the trait of the parents into the forthcoming generation. Means F1 generation, F2, F3. If you want to produce the homozygous plant or the same kind of the plant, or just you wish to retain the same trait into the numbers, you can make the back cross, and especially the test cross is given in the NCERT book. The significance of the test cross. The test cross is useful to know whether the dominant trait is in the homozygous state or in the heterozygous state. For example, what is the importance of test cross? If a plant is there and this plant is having the dominant trait and you don't know whether the dominant trait is in the homozygous state or in the heterozygous state, you will get to know through the test cross. You can say here also, suppose there is a plant and the plant is tall. This is the tall plant. This one is the tall plant. And this tall plant, the tall plant may be hybrid or may be pure. You are not aware that whether the tallness or this tall character is due to the presence of homozygous allele or in the heterozygous allele, or you can say they are pure or they are hybrid. The genotype may be like this also, and this also. As you know, this is the pure tall and this one is for hybrid tall. So to see the plant, you can't say that the plant is pure or hybrid. Morphologically, there is no difference at all in the pure and hybrid plant. If you are interested to know about their genotype, whether this dominant trait means the trait of the tallness, it is in the pure form or is in the hybrid form, after test cross you will get to know. I am going to make here the diagram. You can see the condition first. You take this hybrid plant and cross with the recessive one. What kind of the plants you will obtain? Here the capital T type. Suppose this is the male, this one is the for female. Small t, capital T, small t, capital T, small t, small t, small t, small t, small t. This will be tall, dwarf and dwarf. Ratio, phenotypic ratio 1:1 means 50% tall and 50% is dwarf. One more condition is here, condition 2. If the plant is pure, you can take here the genotype TT, capital T, female, male, capital T, small t, capital T, small t, capital T, small t, capital T, small t, tall and tall. In this case, 100% plants are tall. So here two diagrams I have made. These two conditions I have taken. Condition first, if this plant is hybrid plant, then you will make this cross, because this is, And I told you in the test cross is the crossing of a plant having the dominant trait. Test cross is the crossing of a plant having the dominant trait to their recessive one to know whether the dominant trait is in the homozygous state or in the heterozygous state. Means this is the plant, and the plant is tall, and this tall plant, the tallness is the trait, and you can cross this tall plant with the dwarf one. Suppose this one is the dwarf plant, this is the tall, and this one is the dwarf plant, and you can make the cross. What kind of the cross is this? Test cross. Because in test cross always the unknown parent having the dominant trait is crossed with their recessive one only. And why the test cross you are going to make? To know whether the dominant trait is in the homozygous state or in the heterozygous state, whether it is pure plant or hybrid plant. Through the test class you will get to know. So here the capital T is here. The plant will be tall, and this plant is dwarf. Now there is a question that you don't know whether this plant is in the hybrid state or in the pure state. If you will make the cross, and in F1 generation, if 50-50 ratio comes, if the 50% plants they are going to produce tall and remaining 50% they are going to appear as the dwarf, it means here the trait of the tallness is in which state? In the heterozygous state. It means the plant is what? Hybrid. And condition 2, if the plant is pure, if you will cross the pure plant, this tall plant, with their recessive one, and in F1 generation, if all the plants they are going to appear as the tall plant, it means the trait of the tallness is in the homozygous state. So these two diagrams you have to make to know whether the dominant trait is in the homozygous state or in the heterozygous state. So once again, what is test cross? Test cross is a type of back cross in which a plant having a dominant trait is crossed with their recessive parent to know whether the dominant trait is in the homozygous state or in the heterozygous state. If the plant is heterozygous or the plant is hybrid, after test cross, in F1 generation, the 50% plants they will appear tall, and remaining 50% they will appear dwarf. The ratio is 1:1. So the parent plant having the dominant trait, it will be heterozygous for that particular trait. And if all the plants in F1 generation, after the test cross, they are going to be produced. Here, for example, all the plants they are going to be appear tall. It means the trait of the tallness is in the homozygous state and the plant is pure. So through the test cross, we will get to know that whether the dominant trait in the plant is in the homozygous state or in the heterozygous state. So this is about test cross and the significance of test cross. Now the next topic is incomplete. The next heading is incomplete dominance. Incomplete dominance. Actually, the incomplete dominance, codominance, multiple alleles, Pleiotropy, these headings are given in your book, NCERT book, and all these headings are the result of the deviations of the Mendel's law. Deviations of the Mendel's means exception in the Mendelian principles of inheritance. We have already discussed about the pattern of inheritance as per Mendel's law. These are law of dominance, law of segregation, and law of independent assortment. Except law of segregation, both the law of dominance and independent assortment, these two laws are not applicable in so many plants. In post-Mendelism, some of the scientists, they studied in some of the plants and they couldn't obtain the result as the result which was obtained by the Mendel. And all these exceptions or the demerits, you can say, and these all are incomplete dominance, codominance, multiple allelism, Pleiotropy. All these terms or these phenomena or these process were not given by the Mendels, and these all are the deviations of the Mendels. These all are the exceptions of the Mendels. So one by one we will study about all these headings. The first one is the incomplete dominance. As you know, actually the dominance is of three types: complete dominance, incomplete dominance, and codominance. The complete dominance is actually given by Mendel. His law of dominance, you can say, it was the result of complete dominance. Incomplete dominance, suppose if two alleles are there of a trait, for example, suppose here R and R, these two alleles are there of a character, or you can say of a trait, and suppose the R, the capital R is responsible for red, and this small r is responsible for white. As per Mendel, the red is the dominant trait and white is the which trait? Recessive trait. So you tell me what will be the phenotypic expression, how the plants will appear. As per Mendel, the plant is in this condition, must be having the red flower, because the red R for red flower, small r for white flower, and this red flower is dominant over the white one. So as per complete dominance, as per law of dominance, the capital R is suppressing here the white one, and the plant will appear red, or you can see the plant will have the what kind of the flower? Having the red flower. But in some of the plants, I am going to give you two examples of the plant. There is a plant, 4 o'clock plant. 4 o'clock plant. The scientific name of the 4 o'clock is Mirabilis jalapa. And one more plant is snap dragon. The scientific name is Antirrhinum majus. These two plants are there, 4 o'clock plant and the second one is the snapdragon, or you can say Mirabilis jalapa and Antirrhinum majus. In Antirrhinum majus, in both the plants, in 4 o'clock and snapdragon, in these two plants, incomplete dominance has been formed. What is incomplete dominance? You can see here. In Mirabilis jalapa, or you can see in snapdragon, when the plant having the red flower is crossed with the plant having white flower, in F1 generation, all the plants they appeared pink, F1 plants. You will not write here hybrid. And after selfing, in F2 generation, the plants they appear like this. One you will consider male, one female, or small or red, pink. This will be red, or pink, or pink, and this is small or white. F2 generation plants. So here the phenotypic and genotypic both ratio are same in incomplete dominance. 1:2:1. One is red, 2 are pink, and 1 is white. Now let us understand what is the law of what is incomplete dominance. In incomplete dominance, you can see here, when the two contrasting of a particular trait is crossed, both the alleles of a character, when they are present in the heterozygous form, no one is here dominant, no one is here recessive, and both the alleles, you can see here R and small r, both the alleles, when both they combine together in F1 generation, both they produce an intermediate character, and this is called incomplete dominance. Means, once again, when two alleles they are present in the heterozygous condition, when two alleles of a trait, you can say, when the two alleles of a trait or a character is present in the heterozygous condition, both the traits they produce an intermediate character, and this is called incomplete dominance. Here in 4 o'clock plant and snapdragon, a plant having what kind of the flower? Red flower. And another plant having what kind of the flower? You can see here, white flower. And when these two plants were crossed together in F1 generation, what kind of the plants they appeared? The plants they appeared having what kind of the flower? Pink. Here the parent is red and white, but in F1 generation, all the plants they appeared having what kind of the flower? Pink flower. It is neither red nor white. Means red and white, the capital R responsible for red and this small r is responsible for white. It means the capital R and small r, the red and white, both they mix together and they produce an intermediate character, and that is what? Pink. As per Mendel's, what kind of the flower should be appear here? Suppose here a red flower, capital R is for red flower and this small r is for white flower. As per Mendel law, the flower must be red because capital R is responsible for red and this is small for white. As I told you, as per Mendel, the red is dominant allele and this small white is recessive. So the plant must be red as per Mendel's law. But in these two plants, in 4 o'clock plant and in snapdragon, in these two plants, law of dominance was completely absent, or you can say the law of dominance is not applicable in these two plants, and the dominant and recessive, as per Mendel, here the dominant and recessive one, both they mix together and they combine together and they produce an intermediate character, and due to the combined effect of the red and white, the flower color was pink. And after self-crossing, here the same plant, because the flower is bisexual, in self-crossing in all the F2 generation plants, again the plants they appeared in the ratio 1:2:1. One was red, two were pink and one was white. So here the phenotypic and genotypic ratio both are same in the incomplete dominance. As you have already studied, if you will make the same cross in pea plant, you will get the ratio, the phenotypic ratio, that will be 3:1 and the genotypic ratio will be 1:2:1. But in incomplete dominance, in these two plants, snapdragon and 4 o'clock plant, the complete dominance as which was given by the Mendel is not applicable, and in incomplete dominance the phenotypic and genotypic ratio both are same, and this ratio is 1:2:1. One is red, two are pink and one is white. And this is about incomplete dominance. There may be the question that why the process of incomplete dominance is not said the blending inheritance. Why it is not said to be blending inheritance? Blend means mix. As the parental characteristics, or you can say as the parental traits, they are reappeared. The red and white, these two characters are reappeared into the F2 generation, and that's why the incomplete dominance is not called blending inheritance. If it will be blend in F2 generation, all the plants must be pink. But you can say here in F1 generation, all the plants they are pink. But again after selfing in F2 generation, the parental characteristics, that is red and white, they reappeared. They are not permanently mixed in F1 generation, and they are going to appear in the F2 generation, and that's why the incomplete dominance is not called blending inheritance. It's a very important question as per NCERT. Question is concerned that why the process of incomplete dominance is not called blending inheritance, because the parental traits, or you can say the parental characters, are reappeared into the F2 generation, and that's why it is not called blending inheritance. So this is very, very important. The incomplete dominance is the process, you can say, in which both the alleles, that is dominant and recessive, both the alleles, they produce an intermediate character. Here there is no dominant completely, nothing, no one is recessive, and they produce an intermediate character, and that is called incomplete dominance. And the process of incomplete dominance was reported in the plants like 4 o'clock plant. You can give one more example, a snapdragon. The scientific name of the 4 o'clock plant is Mirabilis jalapa, and the scientific name of the snapdragon is Antihernum majus. You can see your NCERT book, and the scientific names you have to learn. And incomplete dominance is the exception of the Mendel's. In incomplete dominance, Mendelian principle of inheritance or complete dominance is not applicable. So that's all for today, and you please note down some of the questions by the end of this lesson. Please note down question number 1. Differentiate between back cross and test cross. Differentiate between back cross and test cross. Write the importance of test cross. What is the importance, yes, significance of test cross? What is the significance of test cross? Explain with the help of, in the same question write down what is test cross. Explain with the help of diagrams. Question number 3. Name the plants incomplete dominance. Name the plants in which incomplete dominance was reported. Next question. Define incomplete dominance. Define incomplete dominance with the help of diagram and examples. And the last question is, explain how incomplete dominance is different from complete dominance. Explain why incomplete dominance is different from complete dominance. So that's all for today. Tomorrow I will teach you the next topic: codominance, multiple allelism, Pleiotropy, etc. I request you all, I am giving you the assignments every day. and even from the NCERT exemplar I have also given the PDF folder, but hardly I can see the boys they are completing their assignment, as I have to forward your assignment to somewhere that my students they are completing their assignments. I have to keep the record that how many boys they are really watching the videos and completing the assignments. So once again I request you all to complete all these assignments, and you are also supposed to send your assignment in the assignment folder in the Google Classroom itself. So that will be the record for everyone. As I have already given an instruction also, a message that you will log in your account with the folio number and by your name. I hope that you will do as an earliest possible. So thanks all for today. I will see you tomorrow. Thanks everyone.